Good afternoon. Ladies and gentlemen, welcome to the announcement of the 2016 Brain Prize. My name is Kim Krogsgaard and I'm the Managing Director of the Brain Prize. First speaker this afternoon is Professor Poul Krogsgaard Larsen, Chairman of the Board of Directors, who will announce the winners. So, Poul, please come forward. Thank you.
Ladies and gentlemen, thank you very much for joining this session where we are about to announce the 2016 Brain Prize winners. Before the announcement, I would like to extend a deep-felt gratefulness to the scientists around the world who have submitted a great many nominations of very high quality. This collaboration is the very precondition for the prize and for its standing in the international neuroscience community. We are also indebted to the eminent members of our selection committee, chaired by Sir Colin Blakemore. Once again, the committee has dealt with a vast number of nominations. This is not a small accomplishment. Thank you very much. We are grateful for the support shown by the ever-increasing network of international collaborators, individuals, institutions and organizations. Last year, we celebrated the first 5 years of the Brain Prize. Now we are looking into the future. It is our ambition and goal that the Brain Prize will continue to grow in terms of visibility and impact, as will our outreach activities nationally and abroad. Now to the announcement. The Brain Prize 2016 is awarded to Timothy Bliss, Graham Collingridge and Richard Morris for their groundbreaking research on the cellular and molecular basis of long-term potentiation, abbreviated LTP, demonstration that this form of synaptic plasticity underpins spatial learning and memory. Congratulations to all of you. Thank you.
Thank you very much, Poul. And now, next speaker is Professor Sir Colin Blakemore, Chairman of the Selection Committee, who will give us the reasons for this year's prize award. So, Sir Colin, please.
Thank you, Kim, and may I say it's a great pleasure to be here and have the opportunity to speak on behalf of my colleagues about the award this year. Memory is at the heart of our lives. Without it, we couldn't remember the past or plan for the future. Without the ability to remember, we couldn't recognize other people, find our way around in the world, learn to understand language, drive a car, use a smartphone. There would be no education, no literature, no internet. Despite the obvious importance of memory, the mechanism by which our brains store information largely remained mysterious until the work of the 3 scientists who won the Brain Prize this year. In 1894, the great Spanish neuroanatomist Ramón y Cajal was the first to suggest that education, mental exercise, as he put it, might cause enhancement of the connections between nerve fibers, called axons, and nerve cells. Those connections are called synapses. And in 1948, Jerzy Konowski, a Polish scientist who had studied with Pavlov, argued that repetition of activity in nerve pathways might cause permanent functional transformations in nerve connections. He used the word plastic to describe such changes. Plasticity is now seen as a key concept in our understanding of how the brain works, and the crucial discoveries that established the concept of synaptic plasticity were made by our 3 prize winners. The story really starts in 1949, when a Canadian psychologist, Donald Hebb, working at McGill University in Montreal, published an important book, Organization of Behavior, in which he made a prophetic prediction, which has become the most frequently cited quotation in brain research. He wrote, "When an axon of cell A is near enough to excite a cell B, and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, such that A's efficiency, as one of the cells firing B, is increased." Not exactly a poetic quotation, but a very famous one. 5 years later, Ben Delisle Burns, a British neurophysiologist working at McGill, took on a new graduate student, Timothy Bliss. They were interested in memory, and specifically in Hebb's hypothesis that synaptic connections might increase in strength with repeated stimulation. They recorded from neurons in the cerebral cortex of anaesthetized animals and did see changes in the activity after stimulation at high frequencies. But the complex circuitry of the cerebral cortex made the results too variable and too complicated to interpret simply. In 1966, Burns returned to the Medical Research Council's National Institute of Medical Research in London, and Bliss joined him a year later. Tim was much influenced by the findings of a Norwegian neurophysiologist, Per Andersen, in Oslo, who was studying a different region of the brain, the hippocampus, which is tucked under the temporal lobe of the cerebral hemispheres. Damage to the human hippocampus results in a devastating inability to form new long-term memories. And Bliss wondered whether the hippocampus might be, as he put it, the mnemonic engine of the brain. When Bliss contacted him, Andersen told him that one of his graduate students, Terre Lermo, had done some interesting preliminary work a couple of years earlier, but had moved on to other questions for his thesis work. And Andersen said that he hoped that a visit from Bliss to Oslo might persuade Terre to continue the work. Bliss visited in the autumn of 1968 and worked with Lermo, recording from the large pyramidal nerve cells in one part of the hippocampus in anaesthetized rabbits, while stimulating the bundle of nerve fibers that carry information into the hippocampus, terminating on the synapses on the small processes called spines on those pyramidal cells. They found that the response to a single shock was greatly enhanced after a repetitive burst of stimulation, and to their delight, they discovered that a degree of enhancement persisted for at least many hours, perhaps even for a lifetime. This phenomenon, which they initially called long-lasting potentiation, later became known as long-term potentiation, or LTP. In the early 1970s, Richard Morris, who had done his PhD at the University of Sussex, became fascinated with the way in which animals learn to navigate around the world. He was appointed as a lecturer at St Andrews University in 1977 and decided to try to develop a more natural task than the mazes used by most psychologists at the time to study learning and memory. He had the crazy but brilliant idea of teaching rats to swim in a tank of water to find a hidden platform. The Morris water maze, as it's become widely known, has been adopted around the world by basic neuroscientists and pharmaceutical companies interested in memory. In 1982, Morris published a landmark paper in which he showed that spatial learning in the water maze is severely compromised by damage to the hippocampus in rats, demonstrating that this structure is specifically required for that sort of learning. In 1980, the young British neuropharmacologist Graham Collingridge started work as a postdoctoral fellow in Vancouver, in Canada, in the laboratory of Hugh McLennan, a pioneer in the study of transmission of signals across synapses. Collingridge used sophisticated pharmacological methods in the isolated hippocampus of rats to identify different types of receptor molecule in the membranes of nerve cells. These receptors are stimulated by the chemical transmitter substance which is released by axon terminals when an impulse arrives. He showed that the excitatory transmitter substance, D-glutamate, normally acts through so-called AMPA receptors, causing electrical responses from the receiving cell. But another class of receptor, the NMDA receptor, is activated only when the neuron has been stimulated by a repetitive sequence of impulses. By using a chemical agent, APV, which specifically blocks NMDA receptors, Collingridge was able to prove that stimulation of NMDA receptors is essential to establish LTP. Activation of those receptors triggers a sequence of molecular changes inside the nerve cell, which leads to a sustained increase in the strength of the synaptic connection. When Morris learned that Collingridge had shown that APV blockade of the NMDA receptor prevents the induction of LTP, he realized that this might provide a way to demonstrate definitively that NMDA-mediated LTP underpins memory. He found that application of APV in rats prevents spatial learning, but not recall of previous spatial memories in the water maze. Moreover, it does not block other kinds of discrimination learning that are not abolished by damage to the hippocampus. These seminal results were published by Morris and his collaborators in 1986 and 1989. In the past 30 years, there's been an explosion of research on LTP and on synaptic plasticity more generally. In a review article in 2004, Robert Malenka and Mark Baer, two leading researchers in the United States, suggested that LTP might occur to varying degrees and via varying mechanisms at all excitatory synapses in the mammalian brain. Plasticity is now seen as one of the most important features of the brain, influencing early development, implementing all forms of memory and learning, and providing the capacity to reorganize and to some extent recover after damage, such as stroke. The discovery and elucidation of LTP provided one of the starting points for this revolutionary change in our understanding of the brain. It's also had a powerful influence on computer science, stimulating efforts to develop computer architecture based on artificial neural networks. Long-lasting synaptic changes in particular parts of the brain appear to be involved in a variety of clinical conditions, especially depression, neuropathic pain, epilepsy, and drug addiction. And the decline of memory in Alzheimer's disease and other forms of dementia might well involve progressive failure of LTP-like mechanisms. There's considerable interest in the development of treatments aimed specifically at reducing or even reversing this decline. Nobel laureate Edvard Moser has written, "One of the greatest advances in neuroscience over the last half-century is the identification of synaptic mechanisms of plasticity and the role of such mechanisms in reshaping the nervous system during development and in learning and memory." Much of this progress has taken place in Europe. It began with Cajal and Konorski, but in modern times is perhaps the discovery of long-term potentiation and its involvement in learning and memory that has led to the greatest conceptual advances. Thank you.
Colin, thank you very much. Last speaker this afternoon is Professor Jakob Balslev Sørensen for Copenhagen University, who will put this year's prize-winning research into a Danish neuroscience perspective. So please, Jakob.
Thank you very much. This prize comes at a golden moment for the field of synaptic transmission and brain plasticity. Since its discovery more than 40 years ago, long-term potentiation in the different areas of the brain has been at the center of attention in the whole neuroscience field. Scientists have spent a lot of time getting the details just right. This has resulted in a plethora of findings and mechanisms, as well as the occasional controversy. As the prize winners have recently put it, there are many rooms in the mansion of synaptic plasticity. Now we are at a time where the pieces are starting to come together, and with the prize we celebrate that the 3 prize winners, individually and collectively, have established the foundation for our understanding of learning and memory. And we can also start to appreciate the applications for human health that will follow from the seminal discoveries of the prize winners. As we heard from Sir Colin Blakemore, there is evidence that Alzheimer's disease in its earliest form can be due to an impaired long-term potentiation, and with the knowledge of the details of the mechanism, we can start to design ways to counteract this debilitating disease. On the other hand, drug addiction or chronic pain are conditions which seem to be associated with a little too strong long-term potentiation in certain synapses, and the prize winners' work open up new avenues for treating these conditions. So long-term potentiation is one of those rare discoveries in science that seems to gain momentum and importance over time. And I'm sure we have still not seen the end of this process. As a researcher in neurotransmitter release and synaptic transmission, long-term potentiation has been a long-standing inspiration to me. This idea that the synapse is a plastic device means that whenever we do experiments, we look not only for basic function, but we look for changes to this function, we look for plasticity. Some of us look for plasticity at the postsynaptic side, some look for plasticity at the presynaptic side, some of us look for plasticity in the long term and others in the short term, and we make a fuss about these differences in our everyday lives. But in the end, plasticity is the name of the game if we are to understand the brain. As a physiologist, I'm deeply inspired by the findings by Professor Bliss that through basic physiological experimentation at the cellular level, one can discover a mechanism which is of so fundamental importance for the working of the brain. And the painstaking dissection of the molecular mechanism by Professor Collingridge shows me that it is both rewarding and meaningful to look for the molecular basis of physiological processes. Finally, the elegance and simplicity of the Morris water maze, invented by Professor Morris, sets an example for us all when it comes to experimental design. For Danish neuroscience, this brain prize offers golden opportunities for young researchers, not only because they can dream of getting the prize, we all dream of that, and not only because the prize puts Denmark on the map, but because with this prize we attract some of the best minds in neuroscience to come to Denmark. The value of the prize for the Danish neuroscience community is the interaction of these scientists with the local community and the local researchers. This interaction is priceless, so to speak. The prize winners will come not only to accept the prize, but as part of an outreach program, they will attend conferences in Denmark. They will give master classes to young researchers. They actually have quite a program ahead of them, and I hope that they have been properly briefed before they accepted the prize. I also understand that the foundations will make stipends available for young Danish researchers to visit the laboratories of the prize winners and to work with them and learn directly from the best in the field. As a researcher in synaptic transmission, I'm especially happy that my field can now expect a boost from interacting with the prize winners, and I'm also looking forward both to sending young researchers to the prize winners' lab, but primarily to see what ideas they will come back with. At the end, I just want to congratulate the prize winners, Professors Timothy Bliss, Graham Collingridge and Richard Morris, with the prize. And at the same time, I want to compliment the Grete Lundbæk European Brain Research Foundation for an excellent choice of prize winners. Thank you very much.
Thank you, Jakob. Thank you very much. I think that was the end of this session. Thank you very much for your attention. That was it.